Welcome. We're a biology clinic team and we're going to introduce you today about our website, Inventory. Before that, let me introduce our team briefly. This is Ahmed, Mingyu, Kishan, and me, Iris. If you guys want to know about us more, please scan the QR code on the top left corner or visit the link. So let's talk about why we start these projects. Currently in the world, there are so many scientists in the lab that store their data on Excel spreadsheets or still writing on the paper. These are some possible issues arise from this. They have limited capabilities, difficult to search, and so easy to lose their data or organize them. To solve this problem, Jordan's lab at the University of Utah developed their own system using a FileMaker and used that to manage their data. But this software is developed in the 90s and is pretty outdated, have non-user-friendly UI, spend a good amount of money to keep the license of software. So they request our team to develop a new platform to replace their current software. And then let's move on to our demo to show our website inventory.
How's it going? I'm Kishan. I am part of the biology genetics team. And here we can see that we have the first page, which is the login page, and we have the email address and password we can put in. But let's right now make an account. And first we have an email address, username, and let's autofill a password and sign up. And so now that our account has been successfully made, let's log in. And we are taken to the dashboard of our inventory database structure. One of the main things to note is that we do have the strains page, the alleles page, and the plasmids page that can be accessed from our navigation bar. The strains, alleles, and plasmids page are the most frequent databases that are working together, and they have some sort of dependency with another, which we'll go more in detail. But these are the ones that the Jorgensen lab will be working with most frequently, and we will go over the freeze task, thaw task, and import. But if you can see, we have our strains data, but then if you have in our alleles, we have our alleles modifier, but this is not data itself, and we have our plasmids. Now the thing is, we want the Jorgensen lab to be able to import their current data into this database structure. And so what we can do is we can import the strains, alleles, plasmids into a CSV and upload now. Now, because we just created a new user, we do not have admin privileges to edit or delete. And so we can see this here. If we edit it, let's put a sample note in allele, fail to update allele, and we cannot add a new allele as well. So test, fail to create allele. And this is because we are not an admin, as we just created a newly user-made account. So let's sign out, go into an admin account, and sign in. And so now that if you go to strains, alleles, plasmids, and if you go to strains, we can see here, and we can edit our notes. Epsilon strain V7C. More documentation under wiki. And then you press save. The strain is successfully updated. And so now what we want to do is we want to take the data that the Jorgensen lab currently has, and we want to be able to import it into the database itself. So let's do that for alleles, since we don't have any alleles. And we take the allele CSV file, upload it, and now we can see all of our alleles currently here. Now let's do the same for plasmids as well. Here we go to upload now. So now that we have data in our alleles, we can see this is an easy way for the biology database to be able to access their data in this database structure. First, let's start with plasmids. And if we make a plas test, we have all the specific fields that we have worked with the biology lab and made sure that these are the most frequent fields and the required fields to make a new plasmid. So let's just start with position 1, and let's make the date today. and test plasmid for construction. And submit. So now we have our new plasmid. And what we want to do is, let's say we also have the functionality to upload and download a sequence. So let's say we don't have a sequence folder right now, and so we have CGA to replicate a DNA sequence, and we save it. Now let's download, and we get our download as a sequence file, which is specifically the .gb file. Now if I were to erase this, save, we do not have the sequence here at all. But if I were to upload this, we get our sequence back here as we save. So this is one main feature that we worked with to make sure that you are able to download and upload DNA sequences specifically for your plasmid files. Now if we go to alleles, we can see a lot of the main alleles have sort of chromosome types to qualify the data records that we've made. And so we have worked with the biology lab to make sure that these are again sort of the main required fields that are needed for each allele. And if we save, we can update successfully. And here we have all the different headers that we worked with. And so we've also worked with the biology lab to come up with specific headers that they see more frequently. But there are other things such as source, isolation name, and mutation that you are able to select in order to see your full data as well. But let's hide these for now. That way we can see what our biology people are mainly looking for. And so if we go to strains and we go to edit, we can also see that we have our strain name, our position, our alleles, genotype, nodes.
So tech stack, we used Django for the backend since Django would be a great choice to use web framework since they have fast community support that helps a developer while working. Furthermore, Django is a great framework at implementing projects of all scales and sizes, whether it is building a simple website or tackling projects with a large scope. PostgreSQL for database since PostgreSQL is the most capable of all the database in terms of schema support. Front end for React with Vue. At the end, we containerized backend part and front-end part, and eventually we deployed on AWS EC2 T2 micro instance. One of the most impactful technologies harness is efficient cloud utilization. While there are 70 people in their lab now, our sponsor used to spend $2,500 to get 5 people's license by using data storage software, which is called FileMaker. However, by effectively utilizing AWS cloud resource, we were able to reduce the cost from $2,500 to $240 per year. For all of the people in their lab can use our web service. We had weekly meeting with Wayne and Matt throughout our development, leading us to successfully meeting their needs. After the beta test, we met with more people in the lab, Sam, Bertwell, and Nathan, to determine which user case they need more, and we added more features what they asked to us, such as permission, sequence import export, and freeze and thaw request successfully.
To summarize, Genvatory is a tailored solution for genetic research, offering an intuitive interface for streamlined data management. Our team has developed a robust, secure, and efficient database architecture using cutting-edge technologies. Throughout the development process, we maintained continual user engagement, which has been instrumental in driving enhancements to Genvatory. As we conclude, we are excited about Genvatory's future potential and the impact it can make in the field of genetic research. we would like to express our sincere gratitude to Jurgensen's lab for their invaluable insights and collaboration throughout this journey. With Genventory, we look forward to further revolutionizing genetic research. Thank you all for your attention, and we look forward to meeting you at Demo Day.